by acquired DNA mutations that disrupt normal cell processes such as growth, survival, and DNA repair. Key hallmark: growth signal self-sufficiency. This means cancer cells do not need external signals to grow. This is possible because of mutations in genes related to 1. Proto-oncogenes. These are normal genes that can become cancer-promoting when mutated. Example, overproduction of growth factors like PDGF, which lets the cell stimulate itself, autocrine loop, seen in brain tumors. 2. Growth factor receptors. Mutations can make these always on, even without a signal. Example, HER2 receptor overactive in some breast cancers. 3. Signaling molecules, proteins that transmit signals inside the cell. Example, RAS, commonly mutated in many cancers. 4. Transcription factors, control gene expression. Example, MA promotes uncontrolled cell growth. 5. Cell cycle regulators, proteins that control cell division. Example, cyclins and CDKs, when overactive, push the cell to divide without limits. USMLE style questions, 5 choices each. Question 1. A biopsy of a brain tumor reveals cells that are producing their own platelet-derived growth factor, PDGF, which then stimulates the same cells to grow. This is best described as which of the following mechanisms? A. Paracrine signaling. B. Autocrine signaling. C. Mutation in tumor suppressor genes. D. Angiogenesis. E. Hormonal stimulation. Correct answer. B. Autocrine signaling. Explanation. Autocrine signaling occurs when cells produce growth factors like PDGF that act on themselves. This allows tumor cells to grow independently of external growth cues. Why not the others? A. Paracrine involves signaling nearby cells, not self. C. Tumor suppressor gene mutations inhibit brakes, not the gas pedal. D. Angiogenesis relates to new blood vessel growth. E. Hormonal stimulation is endocrine and not cell autonomous. Question 2. A breast cancer biopsy shows overexpression of the HER2 new receptor. This contributes to tumor development by which mechanism? A. Inhibiting apoptosis. B. Increasing autocrine signaling. C. Enabling sustained angiogenesis. D. Providing constant growth signaling. E. Activating tumor suppressor genes. Correct answer. D. Providing constant growth signaling. Explanation. HER2 is a growth factor receptor. When overexpressed, it leads to constant growth signals even without ligands. Why not the others? A. HER2 does not directly block apoptosis. B. HER2 is a receptor, not part of autocrine loops. C. Angiogenesis is separate from HER2's role. E. Tumor suppressor genes are turned off, not on, in cancer. Question 3. A patient is found to have a colon tumor with a KRAS gene mutation. What is the function of the KRAS protein in normal cells? A. DNA repair enzyme. B. Transcription factor. C. Cell surface receptor. D. Signal transduction molecule. E. Cell cycle checkpoint protein. Correct answer. D. Signal transduction molecule. Explanation. KRAS is a GTPase that helps transmit growth signals from receptors to the nucleus. Mutations make it always active, promoting uncontrolled cell division. A. DNA repair enzymes include BRCA1, not KRAS. B. is a transcription factor, not KRAS. C. Receptors are on the cell surface. KRAS acts inside the cell. E. Checkpoint proteins include p53, not KRAS.